with this I come to the end of what lecture? I come to the end of 7 lecture and I will move on to the, okay, we are back to kidney now. What we did so far was this erythropoietin, keep it aside for the time being now. So far we have been focusing on Bowman's capsule and essentially the events that happen there, the flow of blood in, the flow of blood out, the ultrafiltration and the different factors that we have been focused on Bowman's capsule. But what you get there is the filtrate, it is not urine. Okay. For the production of urine it has to be processed and for that it has to go all the way down in the, so Bowman's capsule, proximal convoluted tubule, so here is the Bowman's capsule, okay, and here is the filtrate and it will go here, then it will go into the, okay, let us see, this is the Bowman's capsule, proximal convoluted tubule, okay, and then thick segment and then it, from here becomes a loop of Henle. Follow the story, loop of Henle, thin segment, descending, thin segment, ascending, thick segment, ascending. We will call 1, 2, 3 parts, thin descending, thin ascending and thick ascending. Did you get the 3 names? Thin, thick, okay, thin, descending, ascending, thick, okay. We will call, put together, we will call it as what? Loop of Henle. And as the urine, as the pre-urine flows through all the entire and eventually beyond, it is being processed and leading to the formation of final urine that is ready for it. So what we are going to do in today's lecture is try to focus on how the urine is processed. Now what I will go do is, I will take a, okay, you know this image, this is the Bowman's capsule and from here is the proximal convoluted tubule. You can see the proximal convoluted tubule is convoluted, okay, it goes up and down. Why do you think it's, what may be the rationale in making the tube convoluted? Perfect, because you need to absorb a lot of things, you need to, so it's a good idea to make it. So what I'll do now, I'll take a tiny part of the proximal convoluted tube somewhere here and I'll blow it up so as to show you the different components. And I have, what I have here is the tubular lumen, so this is the filtrate. And the filtrate is going and as it is going it is interacting with the cells of the tubule and the cells of the tubule are single celled, it is single epithelial layer, it is a tube with single layer of cells, okay. And here you have, so we will call them as the tubular epithelial cells and there is a 1 tubular and 2 and 3 and at the, and they are connected to one another by means of what? A special type of proteins which make what you call as a tight junction. What do you call it as? You call it as the tight junction and on the other side you have the interstitial fluid, here interstitial fluid and they have, then we have a capillary. Now what you need to do is, you have to remember this picture because this picture is going to repeat all over as we go along the length of the tube. We are going along the length of the tube and the function of the length of the tube keeps on changing, but it will always be the 3 components will be there. What will you have? The lumen through which the filtrate is passing and it will be lined by the cells which we call as the tubular epithelial cell, the interstitial fluid and there will be peritubular capillary. What do you think will be the pressure in that capillary there? We did it yesterday 3-4 times. What do you think will be the pressure in this? Yeah, you are right. 15, 10, 15, okay. Sometimes it's very low. It's very low. Okay. All right. Now, one more, a couple of terms to be introduced to you. This phase of the cell, which is directly coming in contact with the filtrate, we will also call as the apical phase. What do you call it as? Apical. And the other one I will call as the basal and these 2 phases I will call as, actually there are 4, are you with me? This, if I presume it to be a cube, which it may not be, presume to be, then there will be 6 phases. The other 4 phases I will call as basolateral. I can see basolateral, lateral, basal and apical. Every phase has its own importance, please remember. Apical, basal, basolateral. Are we good so far? Good. This just gives us a summary of the treatment of different molecules and ions that happens as the filtrate travels along the Bowman's capsule, enters into the proximal convoluted tubule so far. Here the proximal convoluted tubule ends and then we have the descending limb of the loop of Henle, thin ascending and then we have this is all and then okay, so let us see what does this K indicate, Na indicate, amino acid, what does it indicate? They are being absorbed. they are being absorbed. So, there is K, Na, amino acid, glucose, water, not much action is happening in this part of the tubule, but then suddenly when it becomes thick, Na, a lot of Na is being absorbed. Here K, look at the arrow, K is in opposite direction, K is being secreted, H ions are being secreted, and finally water is being absorbed. And this gives us, if you take transverse section at different levels, it tells us the how the proximal tubular loop of Henle is extremely thin if you compare the two. Are you with me? And then the collecting tubule will be somewhere here, and the distal convoluted tube, which will be distal convoluted tube, is here, and these are the different, and these will help us to understand as to how the, okay. This is essentially the same figure. Can we immediately identify filtration? So, the fluid is going this way. Are you with me so far? Okay. So, if that is the thing, then this villi that you see, okay, this microvilli that you see is the apical side or basal side? Here. Apical side. Good, good. And then here you have the entire cell, and there is the interstitial fluid, and there is a capillary. In this image, Guyton tries to tell us as to what are the different ways by which the different elements or different ions or can be absorbed. Number one, either the okay. Now here is a cell, and somewhere there is a tight junction. Now, the tight junction immediately gives us a concept that it's tight, nothing should go, but we are wrong. Because although it is tight junction, the molecules are the water molecule is so small that it can go. Sodium ion is so small that it can go. Larger than that can't go. Glucose cannot go. Are you getting my argument? Author is trying to define us what tight junction can and cannot do. Tight junction will allow so water to go, sodium ions to go, yes, chloride ions to go, yes, but no. Okay. Then some of the substance can directly enter into the cell, into the cytosol of the cell, and then they can grow, and then the author tells us about the ATP molecule that makes heart and soul of the whole system. Okay. What are we talking about? Another 15 minutes we are focused on the proximal convoluted tubule. Can you see the diagram there? Can you see the Bowman's capsule there? Can you see the convoluted proximal tubule there? Okay, Guyton has highlighted it. Why? Because now we are just focused where? Where? Proximal convoluted tubule, and that is where we are going to say what is going to happen, and here is the summary. Okay, what we are going to take out of that is sodium, chloride, bicarbonate, potassium, water, glucose, amino acids, and we are going to secrete some hydrogen ions and organic acid. This gives us a little now. Can you appreciate this? What are we looking at? Transmission electron photomicrograph of these tubule cells, okay, section through the proximal convoluted tubule, okay, and this is the transmission, this is the scanning electron photomicrograph. Can you see the villi there? Huge number. This actually should remind you of the brush border when we refer to the intestine. Hello, are you with me? In the intestine we have the brush border, this is exactly brush border. Okay, and the logic is again same, the cell wants to increase enormous amount of surface area, okay, because there are huge amount of proteins there which are selectively doing a lot of function of importing lot of things from the lumen inside this. I will talk about it later, and this gives us a scanning. So, these are all cells, this is the basal side, and these are the actual cells, and this shows the cells in and. Within the cell, which is a very prominent cell organelle that you can visualize. Yeah, you are right. Right. Mitochondria. How many mitochondria can you see? Huge amount of mitochondria. It is a very energy expensive one, because you have to ensure that a lot of ions etc. are transported against the gradient. Okay, so you are going to need ATP, and therefore you have to have a large number of. This is the basal side. What am I showing you? Again, look at this part. If you follow it very carefully, you will see that there is a descending limb, there is ascending limb, and there you will actually find that there is a proximal convoluted tubule and a distal convoluted tubule, and they are almost entangled with one another. They are not really. So, if I take a transverse section, if I take a transverse section here, I am going to get proximal, distal. What is it? What is author trying to tell us? Means, okay, Bowman's capsule. So far, the tube goes down, and then proximal convoluted tubule, there goes 100 times up and down and goes down. Are you okay so far? Then comes up, and then distal convoluted tubule, and the two come very close to one another. and that facilitates transfer from here into the interstitial fluid and then into the distal convoluted tubule. If I take a transfer section there, what am I going to find? The proximal convoluted tubule in transfer section and the distal convoluted tubule in transfer section, but they are very, very close to one another. And on the basal side, okay. Yeah.
That distal is also pretty less in diameter. Is that also significant?
Oh yeah, it is because it's very different. Okay, I'll come to that. We'll come to that. Right now we are focused on proximal. I'll tell you the way these cells handle the fluid that is flowing is very different. Very different. We are still with proximal convoluted tubule. Okay, so we have a section. Okay, so we take kidney of rat. Done. Fix it. Cut frozen sections in a cryotome. Okay, at minus 30 degrees. Okay, and take the sections. Now this time we treat the sections. Earlier time we treated the sections with antibodies against erythropoietin. Now I am going to apply on the sections the antibodies against sodium potassium pump, ATPase. We have done it in earlier semesters. ATPase, hello, yes or no?
Yes.
Great. So we take the antibodies against that, and we find that these are the tubes in section. Okay, and when I cut a tube in section, I find that this bluish part represents the lumen, this part represents the cells, and on the basal side of the cells, I get the immunoreaction. Are you with me there? Look at, so cell is here. Here. The cells are facing this way, that is the apical side, this is the basal side, and these are all the basal. So these are several cells, and on the basal side I get the immunoreaction, which means ATP molecule is located in which membrane of the cells of the proximal convoluted tubule? Basal cell. What cell? Basal cell. Please remember that. And now we are ready to see this cartoon. Instantly you know what I am talking about. What is this? What does this represent? The lumen. What is the filtrate is going down? We are in the proximal convoluted tubule, and the freshly arrived filtrate is going that way. And then these are the cells, these are the tight junctions, this is the interstitial fluid, and this is the capillary there. Done so far? I find that in the apical membrane here, now remember that electron photomicrograph slide, remember those microvilli, okay. There are hundreds and hundreds of protein molecules, okay, which serve as a symport system which will take, along with sodium symport, it will take glucose. Okay, so far? Now why is sodium going inside? Point number one. Okay, let us ask a common sense question. In this fluid, as it is filtered in the Bowman's capsule and flowing along the proximal convoluted tubule, how much do you think is sodium ion concentration in that filtrate?
140.
Absolutely correct. I am so happy. How much it is? 140. But that's in the blood. It's just coming in the blood, and everything, and it is going to be in the same concentration. It can't be more, it can't be less. Okay. Now here, so the concentration here of sodium is 142, and in here how much is this concentration of sodium ions in the cell? 10. Okay, and then here it is in the lumen, it is about if you actually put an electrode there, it is about minus 3 millivolts. Inside is about minus 70 millivolts. Hello, okay, and the sodium is 142 and the sodium is 10. And then you have a carrier there, what is going to happen? With the help of the carrier, the sodium will go in, okay, this is minus 3, inside is minus 70, outside is 142, inside is about 10, 12, whatever it is, okay. So the sodium will drive the transport, and along with that, the glucose will get a transport, it's a import system. So in the cell now you have more sodium, okay. But at the same time, look at the basal side, the side which is towards the capillary. What do we have? We have already seen that in the previous slide. We have that demonstration from the immunocytochemistry protocol. Are you okay there? Okay, so the pump is there. okay, and to make sure that the pump to make sure that every time pump goes through a cycle, means to sodium ions to the outside and to potassium ions to the inside. Every time it goes through a cycle, it will use one molecule of ATP. It will keep on doing that, and as a result of that, the sodium ions in this will be less, and as a result of that, there is a great drive for the sodium ions in this lumen to be taken into the cell. So there is concentration gradient, there is electrochemical gradient, and there is a carrier, so it will go. Just as there is a carrier for sodium and glucose, okay, I will tell you in the next slide, it is called as SGLT. You can immediately make out. S stands for what? GL stands for what? T stands for what? Okay, it comes in that protein system, comes into 1, 2, 3, 4 forms, but don't bother. But SGLT protein, this protein is going to come very repeatedly come back to us. SGLT protein. Okay, it's a carrier system. It is driven by sodium ion. Okay, it is a secondary transport. It depends on the primary transport. The primary transport is because of the pump, which is on the basal side. We are good so far? All right. Just as there is this protein system that accounts for the import of sodium driving glucose, we have other protein systems on a very similar scale, which will along with sodium will drive what? Amino acids, some ions, some vitamins, etc. lot of things. We are just taking one example of sodium chloride, but there are other systems also, and therefore that huge number of what villi, okay, on which the protein systems are sitting. Okay, now if the sodium has gone from here, okay, and then from here it travels, and because there is more sodium here, it just goes passively. There is nothing to drive from sodium into the capillary, but it will be in equilibrium. Whatever goes here, it will be in the equilibrium, and some of the sodium will get into the capillary. Now, it is not at all a matter of surprise that if sodium has gone, chloride will go. Okay or not? Yeah, it will charge, okay, the charge, it will just, if sodium has gone, and if sodium has gone and chloride has also gone, which means osmolality has gone. Hello? Okay, and if the osmolality has gone, what will go now? Water. What will go now? Water. So in this way, so this is how the water goes, this is how the chloride goes, okay, this is how the urea goes, I will study in some details. So the chloride ions take a paracellular transport, which means, so chloride ions being small, how are they going? They are going via the, we are still talking of the same story, but we are going into some interesting details as to, we are talking about the proteins now. Look at this image. Whenever we look at the image, the first thing we get our bearing. Okay, I should know what is north, south, east, west. In this particular case, what do I look for? Where is the lumen? Hello? Okay, so this is the lumen. I know that the urine is flowing this way. Okay, and I know that this is the interstitial fluid, and blood means my capillary is somewhere there. Okay, and I find that in the on the apical side, which is, so I have already seen that. What does it stand for? What kind of SGLT is there? I told you there three are there. Okay, in the kidney, what kind of SGLT do we find? Two kinds. It's a protein system. You know, proteins are notorious for coming in different variations. So this SGLT takes sodium and glucose. Now now we have lot of glucose here, and we want to make sure that we want to absorb every glucose molecule. It's there in the, now this glucose needs to be taken into the capillary. Okay, but glucose molecule on its own cannot really cross the membrane. Therefore, the plasma membrane on the basal side is equipped with another protein system, which I will call as, tell me what? GLUT2. Can you tell me what GLUT or GLUT stands for? Glucose. It's a glucose transporter. What is it? It's a protein system which I call as a glucose transporter, and that glucose transporter, but please remember, glucose transporter has no sense of directionality. Means what? If it's more here, it will take there. If it's more here, it will take there. Get it? It has no directionality, that protein. But as it happens, there's more, because of this SGL, this system, there's more glucose here. Therefore, glucose naturally flow from here to the outside, and from there, if it builds here, then it will pass on into the capillary. So what are we really doing? We are absorbing. Okay, we are absorbing what? Sodium, chloride, water, and glucose. And on similar mechanisms, where are we? Proximal convoluted tubule. Where are we? Yeah, we are still in proximal convoluted tubule. Yes.
Are these GLUT2 channels insulin independent, or they? Or as a matter of fact, are any glucose channel with like facilitate glucose from intracellular to extracellular environment? Do they need insulin like they need for extracellular to intracellular transport?
Okay. As I said, a GLUT will take you from anywhere to more to less, higher concentration to lower concentration. I will focus on two types of cells. One is a neuron and one is a muscle cell or an adipose cell. It's a muscle cell is good enough for my work. Now, you are starving, no food for last 6 hours. As a result of that, the blood sugar level has gone down, it has gone to about 80 mmHg. And when the blood sugar level has gone down to 80 mmHg, your beta cells are almost, the secretion of beta cells has almost come to negligible, which means no insulin is being secreted. And as a result of that, okay, good. Now you have taken food. As a result of that, blood sugar level has gone up. The blood sugar has itself acted on the sensors which are located in the plasma membrane of the beta cells of islets of Langerhans. Okay, and as a result of that, insulin will be secreted. How that will happen, that I will tell you when I talk about insulin. Insulin will be secreted. Now, when insulin will go into the blood, it will act on the insulin receptor which is sitting on the plasma membrane of the skeletal muscle cell. Okay. Once that happens, it will take a signal from the plasma membrane inside the cell, and inside the cell there are inside the cell muscle cell there are vesicles, and those vesicles in their plasma membrane will have V-glutes. Where is it? In the within the cell. Okay. Under the influence of information that is coming from the insulin, the vesicles will now be transported, they will go and be assimilated in the plasma membrane, so that now the glut that was in the vesicles is now exposed to the outside, and now the blood glucose level that has gone up because you had lunch, now that glucose will transfer via the glut now which is in the plasma membrane will go in. It will remain there for 15 minutes. After that glut will again be internalized. Are you with me? Therefore, the transport of glucose across the plasma membrane of a muscle cell and of the adipose tissue is insulin dependent. Good? Go back to brain. Is everybody, it's very, I would have come to this story eventually, but your neurons need glucose every second. Can you afford to let the system remain at the mercy of the glut which is in the vesicles? It will go there, it will lodge, and then no. The brain cells have glutes which are permanently sitting there, constitutionally there. Okay. Therefore, as far as our brain is concerned, the neurons are concerned, glutes are different types of glutes are constitutionally sitting there, so they don't worry. You take glucose, the glutes are there, they will take the glut from the blood into the interstitial fluid, into the neurons, it will be metabolized. Does it completely answer your question? Great. So, let's look at any other question? Okay. Okay. So, look at this. What am I showing you? Just appreciate the beauty of SGLT family. We are looking at what? Yeah, one of them. Okay. Where is it sitting, on the apical side or basal side of those cells? Apical side. If it is apical side of the proximal, this is SGLT of what kind? It's SGLT2. You are right. It's SGLT2. And look at the protein. How many times it goes up and down? 14 times. 14 times. A huge protein molecule. Huge protein molecule. It does what? Import of what and what? Sodium and glucose. Driven by the power comes from sodium. Okay, and then it drives. So this is the author tells us SGLT1 is mainly in the intestine, heart, and kidney. Okay, so the food that we take, carbohydrate digested into glucose from the lumen of the intestine is taken up into the capillaries of the intestine. So as it travels along the brush border of the intestinal cell, what kind of SGLT will be located there? One. Okay, one kind. And then you can read more about it. We again take this section through the proximal convoluted tubule. Sections. There's the same sections. We already have the sections. This time you stain them with antibodies against SGLT. Okay, and therefore so this is actually the lumen of the proximal convoluted tubule, and coming right in contact with the lumen is the plasma membrane of the epithelial cells. Okay, and that membrane is known to have what protein? SGLT2, and you, since you had antibodies against SGLT2, you are labeled. So what you are looking at is SGLT1 here, 2 here, or whatever here. Okay, in this so this kind of immunoreaction you will get. Now that we know about SGLT1 and 2, let's also take time off and try to find out about what. About what? You tell me. You tell me. It's written there. What is it? Glutes. Glutes. Okay. 1, 2, 3, 4. At least 4 different types of glutes are there. Okay. So we are talking about glucose transport, a wide group of membrane proteins that facilitate the transport of glucose over the plasma membrane. GLUT2 is bidirectional transporter. We have already made the point. Transport allowing glucose to flow in 2 directions. It's expressed by renal tubular cells. So in proximal convoluted tubule, what kind of GLUT2 are you going to get? Here it's written, what kind of GLUT is going to get? 2. It is going to be in the, apical side or the basal side? Basal. It's going to, good, it's going to be on the basal side, you are right. This is just a reminder as to, we have seen that on the basal side we have this pump, this protein system, sodium, potassium, ATP. This, I just put this slide to give you an overview of what are the different pumps that you will encounter during the entire tubule. And the one that we have already seen, look at the one, there are 4 points I have summarized for you. What is number 1? What is number A there, please? Sodium, potassium, ATPase. Means these are all pumps, okay? They are going to work uphill, okay, against the gradient, maybe concentration, electrical, whatever, against the gradient, and they are going to use ATP. The second one that you are going to have is what? Hydrogen ATPase. Hydrogen ATPase. Where else do you think in your body you should get an abundance of hydrogen ATPase? Yeah, you are right. You are right. You know something? Our stomach secretes huge amount of HCl. So stomach has proton pump. And what is a proton pump? Hydrogen. Good. Hydrogen potassium ATPase. Okay. Just as there is sodium, potassium, hydrogen, and there is calcium ATPase. Okay. So just remember that there are 4 different types of pumps. Okay. They are all going to use energy, and they are all going to bring about the transport against the